now to Roy Sutcliffe, who's the General Manager at B&K Universal. Roy, thank you for joining us and agreeing to take part today. You have plenty of time to respond to what you've just heard. What do you make of what you've heard on the programme so far?
Good morning. Thank you, Carl. Well, you know, a lot of myths are promulgated by organisations who are against animal research, and you know, it's a myth that animal testing is unreliable because drugs have different effects in people and animals. So drugs that are past the stage in humans occasionally do have serious side effects in people, but during the development of new chemical entities, medicines must pass extensive screening by test tube methods before they're allowed to be exposed to animals. And you know, these tests give valuable information about how the drugs react in living bodies, and they often show up previously unpredicted side effects. For instance, the route of administration may be important. A drug may be given by mouth, or it may be altered by digestion and become less effective or less toxic. And this sort of problem will not be apparent from test tube results, and will only show up in tests in living bodies. The animal tests are designed to reveal potentially undesirable effects such as liver damage or raised blood pressure, nerve damage, or even damage to the fetus. And the results of animal studies will give a strong indication of what effects are likely in people. And it's obviously important, and it's required by law to find out about potential problems before drugs are given to human volunteers and then patients in clinical trials. Human clinical trials involve testing the drug on between 3,000 and 5,000 human volunteers and patients. And if a side effect affects, say, 1 in 10,000, then this could only show up after a long period that the product's been on the market, and it's unlikely that any such problem could have been spotted before. But it doesn't invalidate the research data that's gained from work with animals. The number of drugs withdrawn from the market, it's also consistently overstated by animal rights activists, who often refer to an endless list. In fact, of something like 2,000 types of drug on the market today, less than 40 have been withdrawn in the UK, US or Europe. This indicates a success rate of at least 98% for drug testing procedures. In fact, only 10 of these drugs have been withdrawn in all 4 countries. So the benefits of animals in scientific research are clear. For example, here's a list of the average number of operations performed in the UK each year. So 3 million operations under general anaesthetics developed with the help of animal testing, 90,000 cataract operations, which were pioneered in animal testing, 60,000 joint replacements, 13,000 coronary bypasses. All of these techniques have arisen from animal testing, and advances continue to be made. Keyhole surgery, organ transplantation, skin grafting, and the latest research into the prevention of genetic diseases. They're all benefiting from animal research. And it's certain that any unnecessary reduction in the amount of research would have serious consequences for future research in human illness and well-being. And you must remember that all mammals are descended from common ancestors. One result of this is that humans are biologically very similar to other mammals. Mammals have similar organs, hearts, lungs, kidneys, liver, etc. And they all perform the same functions, controlled by the same mechanisms. And of course there are minor differences, but these are far outweighed by the remarkable similarities.
When it comes down to it, Mr. Sutcliffe, though, most people, if I went out onto the street now with a microphone and said, "What do you think about this?" most people just think it's cruel.
Well, in actual fact, these surveys have been done, and over 2/3 of the British public support the need for animal testing. So I'm afraid that's an exaggeration. And turning to the developments here at Grimston, you know, the site that we occupy, we're not changing the size of the site. The implication is that we're having a massive expansion of the site. Well, we're not. Our development is really a refurbishment and improvement of the buildings within the existing perimeter. And the business has been here for over 30 years, so it's an existing site not being expanded. And yes, we've had some locals objecting to our planning permission, but they're primarily interested in the prices of their houses. And they imagine that if our planning application is refused, their house prices might go up.
I think they might also be concerned about what you're doing to animals as well as the price of their house.
Well, we're breeding animals here. We're not conducting procedures on them.
But you're breeding them for that purpose, aren't you?
We're breeding them for medical research. Yes, obviously.
So you can see why people would be concerned.
Well, the residents of Grimston, in fact, all of them have moved into this area since B&K were already established. So they moved here knowing what was going on.
I appreciate your time today. I have hands up in the studio waiting to come back to what you've said, but thank you very much for your time. Roy Sutcliffe, the General Manager at B&K Universal. Debbie or Victoria, apparently, according to Mr. Sutcliffe, the benefits are clear, and 2/3 of the British public are in support of what's happening.
Well, I'd like to reply to Mr. Sutcliffe and tell him that, first of all, most of the human conditions and diseases which affect humans today do not even occur in animals. So how can you say they are a model which is adequate and equivalent to using for human benefit? There are so many factors and contributors to human diseases that affect humans. These factors do not exist in animals, and they cannot be replicated in animals. I'm talking about factors here like the environment, lifestyle factors, smoking, alcohol, our diet, the exercise we take. How can we possibly use a model based on an animal that does not have any of these factors involved whatsoever and expect to be able to extrapolate results which are going to be of any benefit to humans? Furthermore, I want to bring up the other points that he said about he believes that the public thinks that animal testing is necessary. As Sebastian said earlier, this is no longer an ethical debate. We've moved on from that. Wake up, Mr. Sutcliffe, and wake up to today's world. There are so many superior alternatives around today. We are in an era of unprecedented technological development.
This is where I struggle with the argument, because you say there are alternatives, and he says there aren't.
Well, let me tell you the alternatives. These alternatives have been around for years, and it is only in the recent years with our modern technology and these latest developments that these developments have become incredibly sophisticated and incredibly advanced. And don't forget, let me point out, these models are based on human models, so therefore any results are going to be directly relevant and predictive of the human response, which is just impossible to achieve using animal models. If I can explain some of the examples of alternatives, for example, human cell cultures, human tissue cultures, computer modelling, and microdosing address some of them. If we look at computer modelling, this gives scientists an opportunity to produce a 3D image of the human heart in incredible detail and precision. That it can actually be used to monitor the effect of a drug on that heart, see what happens, why it happens, what could happen, what could go wrong, and what the risks are involved. This can then be replayed at slow motion many times, time and time again, and can produce magnified pictures to show the detail and precision. This kind of method can only deliver results to humans which are far, far superior to the alternative traditional methods, which are just non-predictive of the human response. Why do we have so many drugs today? 9 out of 10 drugs passing these testing based on animals, only to go on and actually fail when they reach humans. We do not know what response they're going to have on humans until they're actually tested on a human patient, and by then it can be too late.
What's happening to, just finally,